My name is Andreas Laustsen. I'm an associate professor at the Technical University of Denmark. There I have the research group called the Tropical Pharmacology Lab. You can find it online if you just Google Tropical Pharmacology. And we are a group of approximately 15 scientists that work with antibody technologies, particularly in the field of toxinology. And I would say right now 80% or so of our activities are within development of antivenoms and diagnostics for snakebite envenomation, for spider bite envenomation and scorpion stings, and even killer bees. So we use a range of different technologies in the lab. One of the prominent ones is phage display. And it's an in vitro, you could say an artificial technique in the laboratory where we can simulate the human immune system so that we can make fully human antibodies without having to inject any human beings with venom or any animals for that sake. So that's one of the key techniques we use to raise these different antibodies that we then use for different purposes. I think you could say our lab is most known for having started by, you could say, pioneering some of the stuff in the field called toxicovenomics, where you analyze venoms and try to split up venoms into the different toxin components and try to figure out not only what is in the venom and how abundant is each component, but also how medically relevant is each component for a human envenoming case. So that was some of the early work we did, and then from there on, I think we, in all fairness, can say, in all humbleness, that we have championed the development of fully human antibodies, where I think we are still the only lab in the world that have done that and have demonstrated that these can be used to neutralize venoms in mouse models. One of the most prominent example is our work with the black mamba, which is a fairly well-known snake. It's the largest venomous snake in Africa, and I think we'll hear more about that in this presentation. And this might be particularly interesting for UK, because I know it's the bite that kills most people in South Africa. So there we demonstrated that we could neutralize the venom using some of the different technologies that we have available. And most recently, actually as late as yesterday, we published, I think, our newest, wouldn't go as far as to call it breakthrough, but at least high impact result, where we demonstrate that not only can we make very good antibodies against different toxins, we can also make very broad spectrum or broadly neutralizing antibodies that can neutralize toxins from different snake species. And I think that being said, I would like to introduce Cecilie Knudsen, who is my PhD student. She has worked with me in my lab for, I think, 4 years now. So we know each other quite well. And so Cecilie started as a young master student, did her thesis work in my lab and had extremely good results, and has been forced to go to Costa Rica and Cambridge University and a few other places throughout these years, and is now stuck in the lab with me and particularly working on Snakebite Diagnostics, but she's previously been involved in therapeutic research as well. And I thought it would be nice to give her the opportunity to present some of the work that we've been doing, because she's actually been involved in many of the projects that we've had ongoing in the lab. And in my opinion, she's a fantastic presenter. So I think without further ado, I will hand over the word to you, Cecilie, to take us through the presentation. And I think it would be good that, and I can see everyone except me and Cecilie now have our microphones on mute. I will mute myself in a bit. And then if any of you have any questions, write them in the chat. I will maybe take a few notes, and then we'll end up with a Q&A session where we can try to answer your questions as good as possible. So with that, Cecilie, the word is yours, and thank you all for showing up.
Right, thank you, Andreas. Perhaps you can help me with the questions, because I can't really see them as they pop up. So, as Andreas was saying, we're going to talk about snake venoms, antivenoms, and diagnostics. And to start out, let's talk a little bit about the problem of snakebite envenoming. Every year, more than 5 million people are bitten by venomous snakes around the world, and roughly half of them become envenomed. Out of these, more than 400,000 end up with permanent sequelae, and about 400,000 end up with permanent sequelae. and more than 100,000 die. So what are these snake venoms that can have all these horrible effects on humans? Well, snake venoms can consist of more than 100 different toxins, and for simplicity's sake, they're often grouped into three different categories: chemotoxins, cytotoxins, and neurotoxins. And each of these categories of snake toxins can affect the human body in different ways and give rise to a distinct clinical syndrome. Just a brief warning, there is an unpleasant picture on the next slide. So looking at the first of these syndromes, local effects, often caused by cytotoxins, you can see clinical manifestations such as on this picture, where this child displays very characteristic skip lesions of necrosis. You have degradation of muscles, you have pain and edema. Those are all very typical for these types of toxins. Different clinical syndrome is hemotoxicity. So that can result in shock, it can result in the blood coagulating, but most frequently actually it results in the opposite, that the blood loses its power to coagulate. So to look a bit further into this, I have this video, which hopefully is also on display on your screens, of these gentlemen milking this venomous snake. So the way you do that is you make the snake bite through a membrane and inject its venom into a container, which is seen here. They then take this venom and add it to a blood sample. And here we can see that the venom has made the blood coagulate. So to understand what's going on, it is useful to first know a little bit about how coagulation occurs under normal circumstances. And that is through the blood coagulation cascade, the last part of which is shown here on the left. And for today, at least, it's not important that we understand all the small details of it. We just need to know that it's essentially a bit like a set of dominoes where you have one factor which activates the next, which activates the next, down to the point where prothrombin is activated to become thrombin, which can then cleave something called fibrinogen into fibrin. And fibrin is a bit like some, you could say, long sticky threads. They can form a bit like a sticky spider web that can catch different blood components and in this way form a clot. So that's what happens when the blood coagulates. In the body, this is counteracted by something called plasmin, which then dissolves these clots. So what some chemotoxins can do is that they can go in and they can work just like thrombin does to cleave fibrinogen into fibrin. And you would think that this would result in the formation of blood clots, but actually what happens in an envenoming scenario is often that that the plasmin will dissolve all of these blood clots down to the point where all of the fibrinogen is depleted and the blood loses its ability to coagulate. And this thing with the snake venom toxin going in and acting very similar to something that already exists in the body and which has a normal physiological function and which is usually good for us is actually not very unique. That's something that happens quite often. For example, there are toxins that can mimic what happens at pretty much every single point in this blood coagulation cascade and sort of make this whole system go haywire. Similarly, we have toxins which can affect, for example, angiotensin, which is part of regulating our blood pressure, and toxins which can go in and sort of make our blood vessels leaky, making us more prone to internal bleeding. So we talked a little bit about cytotoxins and now about hemotoxins. So the last syndrome we're going to talk about is that caused by neurotoxins. So what you can see here, or should be able to see at least on the right, is a video of a mouse which has been injected with neurotoxins from the black mamba. And this mouse has lost control over its muscles, so it's having all of these involuntary muscle contractions, spasms, if you will. While that can be quite typical of neurotoxicity, that's not what usually kills you. That's actually kind of the opposite, because with neurotoxic snake venoms, often you eventually have onset of paralysis, often something called descending paralysis, which sort of starts at the top of your body and then works its way down. And by the time it reaches your respiratory muscles, you lose the ability to breathe, and then you die from asphyxiation if you don't receive some form of help. And of course, if the venom dose is adequate. And again, just to briefly go into how these toxins work, here we have a classical textbook representation of a synapse. So you have, I don't know if you can see my mouse if I do this, but you have the presynaptic region here. So usually what would happen would be that you would receive some sort of signal transmitted down through the neuron, down here to the presynaptic area. And if this signal is strong enough, you will have release of neurotransmitters here into the synaptic cleft. And on the postsynaptic membrane over here, that could be on another neuron, it could be on a muscle, you have these small receptors which can then detect the presence of these neurotransmitters. And when they do, they can propagate the signal here. For example, a signal that a muscle needs to contract. Similarly to the hemotoxins, we have neurotoxins which can interfere with the system by mimicking something that's already part of the system, in this case, the neurotransmitters. So we have toxins which can go in, and because they're kind of similar to the neurotransmitters, they can occupy the binding sites on the neurotransmitters receptors, thereby making the receptors unable to detect the presence of the actual neurotransmitters, and therefore unable to propagate a signal telling the muscles to contract. And that's what happens when you're paralyzed by at least some of these snake venom toxins. And again, there are neurotoxins that can interfere with pretty much every single part of this neurosignaling cascade. Fortunately, all of this stuff doesn't have to happen. There is a solution, and that solution is antivenom. Antivenoms today are frequently produced first by milking a snake or several snakes, just like we saw in the video. You then use this venom to immunize an animal, often a horse. Immunize in more daily terms is also called vaccinate. So essentially you vaccinate a horse or another animal. And the animal's immune system will then produce antibodies. These antibodies are present in the blood of the animal. So you can take blood from the animal, extract the antibodies, give the animal the rest of the blood back so it isn't harmed. And then these antibodies can be formulated as an antivenom capable of neutralizing snake venoms. So that's just perfect. Or is it? Well, antivenoms are invaluable. They save lives. They're incredibly important. But there are different disadvantages associated with them. For example, it is true to varying degrees for different antibodies, but it has been shown that in some cases, the worst cases, as little as 5% of the antibodies in an antivenom actually neutralizes snake venom toxins. And the rest of the antibodies, well, they just recognize something completely different, whatever the horse has been exposed to previously in its life. And of course, if only a low percentage of the antibodies in an antivenom recognizes and neutralizes snake venom toxins, well, then you need a larger dose to neutralize a given amount of venom, compared to if all of the antibodies had been capable of neutralizing the toxins. Another problem is that these are animal proteins that we're injecting into the human body. And there is a risk that the body will recognize these horse antibodies as being foreign, and that the patient receiving the antivenom can have very serious allergic reactions. So in that sense, there can be a health risk to injecting antivenoms. Other than that, producing antivenoms in this way, there may be batch-to-batch variation. No two horses are going to produce exactly the same antibodies. There's also an issue that these antivenoms can be quite expensive, and this is especially problematic because snakebite is associated with poverty. A lot of people who are bitten by venomous snakes are poor, and if they can't afford the treatment that could potentially save their lives, that is hugely problematic. And this expense is also tied a little bit to the fact that antivenoms have a fairly long production time. I assume all of you have been vaccinated against something at one point in your lives. I think personally the last thing I was vaccinated again was hepatitis A and B. And that process took about half a year and multiple shots at the doctor's office. And it's kind of similar for these animals that are used to produce antivenoms. They need to be to receive multiple doses over long periods of time. So sometimes it can take more than a year to produce an antivenom. And that can be problematic if there is suddenly a surge in the need for antivenom there, which has happened. For example, there was this case with a valley being flooded that flushed out a lot of snakes. So there was a spike in the snake bites, but you simply couldn't just magically make more antivenom appear. You had to wait to produce more. And by the time that was possible, I mean, these people would either have succumbed to envenoming or survived anyway. And the combination of all of these factors means that often antivenoms are not available where they're needed. And this is a problem that in our laboratory, we really hope to help alleviate. And the way we mean to do that is producing an entirely novel type of antivenom, one based not on antibodies produced in horses, but on human antibodies produced in a lab. So our belief is that by doing this, by sort of engineering these antibodies, we can make it so that 100% of the antibodies in an antivenom have been proven to neutralize snake venom toxins. Because these antibodies will be human, they won't have the same potential for provoking side effects and allergic reactions. They'll be produced in the lab, which is a lot more controlled than in a horse, so we can be more certain that we get the same product every single time. There have been made preliminary calculations showing that these antivenoms using human antibodies should cost about the same as current antivenoms, or hopefully less. But this is something that remains to be proven out there in the real world. These antibodies produced in the lab, once they have been developed, can be produced in a few days. So that would really alter the time scale compared to current antivenoms. And hopefully these properties in combination will help make this novel type of antivenom more available. So we started out by meaning to make a prototype antivenom against the venom of the black mamba. But just how do you get human antibodies against black mamba toxins? Because immunizing, vaccinating, it's not such a good idea, and it's also not legal at all. So we had to figure out a different way of doing this. And to do so, first Andreas and later I went to Cambridge to a small company called Iontys. And the reason we did that is that at Iontys, they have a collection, a so-called library, of lots and lots and lots of different human antibodies, 4 times 10 to the power of 10. So in theory, in this library, there's an antibody recognizing pretty much everything. The trick, of course, then is to find the few antibodies in this huge collection that are capable of not only binding but also neutralizing snake venom toxins, and that's a little bit like finding a needle in a haystack. So the way we do this is using a technology called phage display, the inventor of which actually just received the Nobel Prize last year, so that was super exciting. And phage display roughly works like this. So you have a test tube, shown here as this kind of white box at the bottom. And we can then immobilize the snake toxin we want to find antibodies against inside this test tube. So the toxins are stuck. Then we add our collection of all of these many different human antibodies. And the antibodies that can bind to the toxins are allowed to do so. And then we can pretty much just pour the rest of them down the drain. So just, tip the test tube upside down and they fall out. So that's one cycle of this phage display selection. Add all the antibodies, allow the ones that can bind to bind, wash everything else away, and then you're left with something that binds. And what we usually do then is we make more copies of them, and then we start over and repeat this process just to narrow down the field until we're left with only the very best antibodies. So having done this, I went to Costa Rica to Instituto Clodomiro Picado, where they produce antivenoms but also have a lot of research in there. A bunch of amazing people doing fantastic work. And the reason I went there was to test a new prototype antivenom. So doing this, of course, we had to start with a control. So we injected black mamba venom into the brains of some control group mice. And as you can see from this graph, we have survival as a function of time. These mice died, unsurprisingly. The real question, of course, was, can we save these mice from dying if we also administer them with our prototype antivenom? So we pre-incubated venom and antivenom, that is, we mixed them, we left them on the table for a little while, then we injected them into the brains of these mice and monitored if they survived or not. And amazingly, actually, at the dose shown here, all of the mice survived, and that was great. There was Eye of the Tiger blasting in the background, everybody was happy. But naturally, this is only a preliminary assay, and there are some differences in this assay to a real-life situation. For example, the fact that we chose to mix a venom and antivenom before injecting it into the mice. One reason we did this is that it's actually completely standard procedure. If the antivenom does not work in this very mild scenario, there is no reason to go on to test it in a more lifelike scenario where you first inject the venom and then, after a delay, the antivenom. But since our antivenom prototype worked in this scenario, that is something we can now move on doing. Similarly, we gave these injections into this fluid-filled compartment of the brain, the cerebrospinal fluid. And that's also not how envenomings tend to occur in real life. You would at least need a very surgical snake to inject venom directly into the brain. So next up, to make this even more lifelike, is we would have to inject the mice with venom intramuscularly or subcutaneously perhaps, and then afterwards inject the antivenom intravenously. But nonetheless, this preliminary test showed promising results. So we've kept working with this. But something else we're also working on is finding antibodies that bind not only one toxin from one snake, but actually multiple toxins from different snakes, so that if you don't know which snake bit you, well, these antibodies in this new antivenom could actually just neutralize regardless. And the majority of the work I'm showing here has been carried out by Andreas and my colleague Sharon, seen at the top left. And the way we do this is, so I told you before how we immobilize toxins at the bottom of these reagent tubes, add a collection of antibodies, let the ones that can bind, and wash everything else away, and then we repeat. Usually when we repeat, we just use the same toxin again to find antibodies that are even better at binding that specific toxin. What Sharon did here was she changed the toxins when she repeated. And in this way, she actually selected antibodies capable of recognizing multiple toxins. In this case, cytotoxins from these cobras shown on the right. So here we have a preliminary result from her. 24 different antibodies tested against cytotoxins from different snakes, shown with these bars. And we can see all of the antibodies marked here with stars actually recognize at least 3 different cytotoxins. Should maybe just say, so we have here cytotoxins from Naia nigricollis, Naia mozambica, and Naia melanoloca. And as a control, we have a completely unrelated neurotoxin from the black mamba shown in orange. Next thing we did was we wanted to see if, other than just bind to these toxins, if these antibodies could also neutralize them. And we did that in a cell-based assay. So here we have 4 graphs. If you look at just any one of them, at first you can see a control that's just cells doing fine, surviving on their own. Next bar, you can see what happens when we add cytotoxins to those cells. They start dying off at a rapid rate. If you add our antibodies, nothing happens, the cells are fine with that. And then the 2 last bars are actually what's most interesting. So here you can see what happens when you add a mixture of toxin and antibody. And we see that these antibodies can actually neutralize the lethal effects of the toxins, and the cells start surviving in spite of the presence of the toxins. And this happens for multiple different cytotoxins for different cobras. So this is some of the newer stuff we're working on, and it's what was just published yesterday, and I'm sure Andreas will be happy to elaborate on that after the talk if there are any questions. But this brings me to the final part of my talk, because as it is right now, antivenoms only work when they match the venom. If you're bitten by a cobra and you receive rattlesnake antivenom, it's not going to do you any good. As we discussed previously, it might actually do you some harm. So you need to know which snake bit you in order to know which antivenom you should potentially receive. And that can be quite difficult. I realize that I'm talking now to a group of herpetologists, so maybe for you this isn't as difficult as it is for everybody else, but many people at least have a very hard time identifying which snake bit them if they even see the snake at all in the first place. And here on this slide, I have a classical example of one snake which has a very potent neurotoxic venom and another one which is not venomous at all. Here I have a trickier example. So some of these snakes belong to the same species, others don't. And in the interest of time, I don't think I'm going to have you tell me which is which. I'll just show you. But this again just goes to show the amazing color variation that can be seen in these species, which also underlines just how difficult it can be to figure out which specimens belong to the same species. These are other examples, also all the same species, but very different coloration. So if you can't just look at the snake and recognize it, what do you do as a doctor when you have to diagnose a patient? Well, typically you talk to the patient, figure out, okay, did they see the snake? Did they perhaps take a picture of it? What time of the day were they bitten? What happened? But other than that, you monitor the clinical manifestations of envenoming. So we already talked about some of those. Could be that the patient has incoagulable blood, as seen here on the left. It could be that they have signs of paralysis, as seen on the top right. This guy's right eyelid is beginning to become paralyzed, so that's a typical sign of descending paralysis, which might eventually reach his respiratory muscles if nothing is done. At the bottom right, you have different urine samples with varying amounts of blood in them. And a skilled physician can sort of look at these different clinical manifestations and use them to narrow down, okay, these manifestations and a bite in this area this time of day, that is suggestive of this or these species of snake, and therefore this type of antivenom might be warranted. That being said, this can be very difficult to do, especially for doctors who are not very familiar with treating snakebite envenoming. So a final project we're working on in our group is trying to figure out an easier way of telling which snake bit a patient, and hopefully through this being able to administer antivenom earlier, since early antivenom administration has been associated with better patient outcomes. And pretty early on we realized that maybe what we need is actually not the most sophisticated, sensitive diagnostic test out there. Maybe it's more important that whatever we come up with is cheap, because again, snakebite envenoming is associated with poverty, and if nobody can afford the solution, it doesn't really help anybody. Whatever we come up with also has to be fast, because it's not really going to be useful to be able to tell the snakebite patient 3 days after the fact, oh, it was this snake, because, they should probably have received treatment already by then. And finally, it's important that this method is efficient and robust, preferably very easy to use so that people can use it even if they haven't tried using it before, and so that it's stable across a wide range of temperatures and humidities. And a method, a technology that came to mind that seemed to fulfill all of these criteria are lateral flow assays, also known as the type of assay seen in over-the-counter pregnancy tests. They're cheap, fast, take 5 to 15 minutes, and they're sort of very user-friendly and robust. So we were imagining something that works a bit like this, with a type of barcode, where if one line lights up after you take a blood sample from the patient, it could be a rattlesnake. Second line might be a lancehead. Third line could be a bushmaster. And of course, there's a control line to verify that the assay has worked as intended. And I think that's actually all I have time for. So if there are any questions, Andreas, maybe you can help me answer those.
I don't think there's been any questions written in the chat, so people are either welcome to write them now or simply just unmute themselves and ask questions. But maybe I can ask the first question to you, Cecilie. So how is it that your work with antivenom, how does that technically relate to these lateral flow assays? Because we also use antibodies there.
Yes, so you could say that it's very similar in the sense that both for the antivenoms and for the diagnostics, we're really just working with finding antibodies that bind toxins, and very particular toxins. So in the case of Sharon's work, for example, just as many toxins as humanly possible. And in my work, I want antibodies that very specifically bind to toxins from if I want to diagnose a species, then only that species. Right now, though, I'm working on diagnosing groups of species, for example, all rattlesnakes. Well, then I want toxins that recognize something present in every single last rattlesnake venom, but nothing that's present in the closely related venom from the other snakes present in the same area.
So maybe a different question. So now we've been talking about snakes. Could you maybe, Cecilie, talk a little bit about how bad of a problem is scorpionism or spider bite envenomation in comparison, at least seen from a human perspective?
Well, to be completely honest, I don't remember the numbers. Maybe you do. But snakebite really is a bigger problem just looking at the number of cases, and I think actually also at the severity. But maybe you would prefer to discuss that yourself.
I mean, I think for spider bite, the numbers are truly unknown. It is known that some spiders, particularly in South America and Australia, are lethal to humans. But in reality, the true numbers of how many people die or at least are harmed by spiders are not known because they're typically not recorded. Scorpions, I think it's estimated that a few thousand people might die per year, so that's a lot less than snakebite. But I think that in some regions of the world, scorpions are actually a larger problem.
Actually, I think in Brazil they have a huge problem with one particular species.
Yes, and also in Mexico, I know that now we have people from Mexico here, that there are some areas where people are stung multiple times throughout their life. And even though the people don't die, they may suffer quite a bit for several days, to an extent where antivenom is actually needed. But part of the problems that you presented earlier, Cecilie, with antivenom causing these potentially severe adverse reactions, that you could say disqualifies them from being used repeatedly, because the human immune system builds up a strong immune response against horse proteins. So you might use the antivenom once, but even the second time, your body is even more geared towards launching a hyperallergic reaction. So that's also one of the problems, and the problem even is that if you've been stung by a scorpion and you receive antivenom, you will actually not really be eligible to receive antivenom in case of snakebite, because it's the horse proteins you act against. I can see that I think Roberto has a question whether we are getting antibodies for the most toxic fractions or against all mammal lethal components. That's a very good question. So I think in terms of, we are going for the most lethal fractions for humans, and we are, generally speaking, trying to find antibodies against every single toxin that is present in an amount where it could kill a human being. Some toxins would be lethal, if we talk about mammals in general, in the abundance they're present in for mice. To not overcomplicate things, we do not particularly search for antibodies against those. But the thing is, there are a few exceptions, but most toxins that are harmful to mice, or the same toxins that are harmful to humans. So despite that we focus on human envenomings, our antibodies could actually also be useful even to protect mice if that was the case. That's also why we can do a lot of these experiments in mice. And not always is the preclinical model representative of the human disease, but in case of snakebite, it's fairly comparable to an extent where it's predictive enough to do it in mice to see this is likely or more than likely to also work in man. So Cecilie, maybe looking a bit into the future, how far into the future do you think that we have to go before we see some of these newer therapeutics that we've been talking about, or even diagnostics? How fast could it go if funding was not a challenge, and how fast do you think it'll go? Do you think that we will have horse-derived antibody-based antisera or antivenoms 50 years from now?
Geez, I feel like I'm at an exam. I think it's a really interesting question, and I think, honestly, there are two things to it, right? Because one thing is how fast can science move, and we can make something that works, I'm convinced, fairly soon. Something else is getting it tested and approved so that we know it's safe to release it for use. And I really think clinical trials and clinical approval is actually what's going to take the most time. And something else is also sort of, I guess, in a sense, the willingness to change. And I don't necessarily believe that it's an either-or situation. I mean, we have at least both participated in discussions where it's been, well, yeah, discussed that maybe one way to do it, at least for starters, is that to supplement current antivenoms with human antibodies. Because current antivenoms, the way they're produced, you get antibodies against the most immunogenic components of the venoms. The most immunogenic components often just being the largest components, not necessarily the most lethal components. And you do have examples of venom toxins which have immunomodulatory properties that mean that they can sort of evade the animal that's being vaccinated's immune system, so that it's just really, really, really difficult to produce antivenoms against using these traditional methods used for antivenom production. So maybe for starters it could be a combination where you have these antivenoms and then you just spike them with human antibodies that recognize these tricky toxins before eventually perhaps transitioning into something based
so we're basically talking about, you could say, hybrid antivenoms where we fortify existing antivenoms with some of these antibodies to overall lower the dose and make them more potent.
Yes.
And I see there's a comment from Mike who says that in many cases when you administer antivenom when it's needed, that anaphylaxis can be dealt with. And that is, of course, that's true. But it does, to an extent, require that you have typically a tertiary healthcare center. It's difficult to deal with anaphylaxis in the field. It's difficult to deal with en route to the hospital. I'd say, of course, neither me nor Cecilie are medical practitioners, and we haven't treated snakebite ourselves, so our knowledge of this is based on what we hear from our collaborators. But what we understand is that it is very uncommon that you would administer antivenom before you actually reach a tertiary clinic. And we had a case with a student in our own group who had to go to different clinics, had to be referred despite needing antivenom, simply because the first places he went, they were not equipped to deal with anaphylaxis. There was no necessarily, they weren't sure that he would have an anaphylactic shock, but they were so afraid that they preferred to transfer him several hours away. And in that case, it's, you could say, at least beneficial in allowing antivenom to be administered earlier if you can reduce the risk of these immunogenic effects.
Yeah, I've also been interviewing some snakebite doctors in Brazil who've told the same story that, well, if you have the necessary equipment to deal with it, it's not so big of a problem, but often, especially in the remote areas, they simply don't, and that makes them delay antivenom administration when they would otherwise have gone on with it.
Yeah.
But of course, very, very area specific.
Yeah, true. And there's another comment from Kay about why, well, not why, but he noticed that a lot of the snakes that we've been working on are from Africa, and there's a few good reasons for that. One is, of course, that there is a significant snakebite problem in many African countries. It's also one of the places that not all countries, but some. I think South Africa is known for having a fairly good system for dealing with snakebite, both you could say, if you look at treatment as a whole, but it's a place where the snakes are very venomous, there are many bites, and many countries lack access to antivenoms. So you could argue many North American snakes are also very dangerous, but there is antivenom, maybe not most, but many places where prevalence is high. So there are also other places like India and Southeast Asia where snakebite is indeed also a very serious problem, and some of these snakes we also look at. Then Mike says that it's key to keep cost low, and I completely agree. It's, I think, often an overlooked issue, not just in snakebite, but for many diseases, that cost is something, especially for some regions of the world, that prevents patients or victims to have access to medicine. And snakebite is maybe particularly one of these cases, where we have examples from North America where antivenoms are prohibitively high unless you are insured. You have the same case in some African countries, at least to my understanding. And I mean, as I think Cecilie also mentioned earlier in her talk, it doesn't help anyone if your perfect solution is unaffordable. So I think we've been talking about safety is important. If you can increase safety, you can hopefully administer treatment earlier. Efficacy in itself is important, but existing antivenoms are, if they are produced in the correct way, also quite efficacious. But really what is, in some parts of the world, keeping patients from being treated is cost. And it's something that's difficult to deal with because it has less to do with science, more to do with clinical development and manufacturing. But it is something to an extent that our group has at least tried to address through looking at relevant biotechnological methods for manufacturing antivenoms and trying to provide, you could say, prospective cost estimates. And I think next week we expect there will be an article in Frontiers of Bioengineering and Biotechnology where we try to estimate different types of antibodies. How much would it cost to use these individual components to formulate, you could say, a new type of human antibody-based antivenom. So clearly it's unfortunately never going to, I doubt that it's ever going to be so cheap that you can just treat people for 1 cent or even just $1, just filling a sterile vial without, even if it's just sterile water, just filling the vial in Europe costs €50 in itself. In most countries it's at least above $5. So just the whole handling of a pharmaceutical in liquid format comes with a minimum cost. And producing antibodies has become dramatically cheaper in the last 30 years, going from being unaffordable in almost every country to now being at a more reasonable level. So I mean, I think we can, there's a limit to how low you can go, unfortunately, but we can definitely do better than what we have on the market now. I think surely in the future, but it's not going to happen overnight. So the question about the idea about, so that's Masood having a question about how long the half-life of new antivenoms would be. So that has less to do with actual stability and more to do with regulatory affairs and what is the standard in industry. And it's uncommon that any medicine has an official half-life of more than 2, sometimes a little bit more than 2, but it's not common, it's more than 2 years, despite the fact that if it's a Panodil or some aspirin, surely you can keep it for 10 years at room temperature and it will still work. It's just legally not approved for that. I have tested existing antivenoms that have been on a lab bench in tropical climate. They've been standing for 8 years. and they still do the job. They can still effectively neutralize venom. So it's more sort of an extra safety precaution that you don't want things to not be refrigerated, not stay out for too long, because over time there is of course a little bit degradation, and what you see is that the safety profile gets worse. You see that some of the proteins and antivenoms, they will aggregate, and they will, when injected, have a higher propensity to cause some of these potentially anaphylactic shock, but serum sickness and other immunogenic reactions. So technically the shelf life of a new antivenom, if you thought about using some of the newer antibody formats such as nanobodies or DARPins, and there's a range of other types of binding proteins, realistically you could have a functional shelf life, I think, of at least 10 years. But it's not only what is technically feasible, but also what regulatory affairs will approve. And that is really dependent on each and every country's regulatory system. So it's not so easy to answer just as a scientist. But if there are no more questions before I will let Cecilie, I think, have the last word, I would just like to say thank you so much, all of you, for showing up and showing your interest in both Snakebite and Venom itself, but also hopefully a bit in the work that we're doing in the lab. It's been a pleasure, now also mainly listening to Cecilie, so that makes the pleasure larger, but thank you so much for showing up. And if you're interested, please reach out, as Masoud said. Cecilie?
Yeah, I also would like to thank you for your attention and thank Masoud for the invitation. And as was on the last slide for a couple of seconds at least, if you think of any questions, there are our emails addressed, so you can reach us. Yeah.
Yeah, so the question about how to make antivenom cheaper. So I think when Cecilie and I talk about, you could say, these next generation antivenoms, they are manufactured independent of snake venom. We use, of course, snake venom to develop them, but once they're developed, snake venom is not needed for manufacture. Existing antivenom production, I have to say, it's not very clear to me how you really make it a lot cheaper, because it is something, at least where the good laboratories have done their best in the last 50 years at reducing prices, in how to keep and maintain horses or sheep. I'm sure you can, to some extent, by optimizing your stables, optimizing your serpentaria, that surely will help. It will help to have fewer people, so less labor costs, but there is a maximum of serum you can get from one horse, and the cost does actually sort of just scale by multiplication when you go up. So there is a bit of a benefit to having a larger serum institute, but it is hard to get it very far down, because you do need, for every you could say number of vials, you do need an extra horse, and these are just very expensive to keep. So this is also why you could say that we believe that if you go into the future, if you do change from using animals for manufacture and can change into fermentation technologies, there is a chance that we can dramatically reduce prices, because that does scale a lot better and allows for cheaper production, which we know from insulin production. We know this from also other antibodies within cancer research. We know it from industrial enzymes. We know it from blood factors and many other biopharmaceuticals.
Okay, doesn't look like we have more questions. People are saying thanks and appreciate and so on, and very interesting. Of course, it was a very interesting presentation. Well, thank you very much, Doc and Cecilie. I hope you good luck, and hope the school opens and the labs open and you can get in there and finish up everything. Really enjoyed and appreciate for spending time with us.
Thank you, Masoud. Take care, everyone. Have a nice either day or night or wherever you are.